ladies and gentlemen. As you know, yesterday we reviewed the structure of DNA and how amazing DNA was as a molecule. We talked about the fact that you are made of approximately 40 trillion cells. And of those 40 trillion cells, there are 200 different types of cells. But every single cell in your body has the exact same DNA. Amazing. Then on top of it, 99.9% of our DNA is identical, and only 0.1% is different. And that 0.1% difference allows us to do things that we're going to do today, like our gel electrophoresis lab. Normally when you're in this class, you are biologists, but today, you get to be DNA technicians. As you read last night in your homework, in your pre-lab, you read a background of a crime scene, and you guys are going to be using gel electrophoresis and DNA fingerprinting to determine who the culprit was of this crime. It is not often that we get to experience such labs, so I want you guys to appreciate how amazing this experience is and how much I love this lab, as I've told you before. Almost as much as I love the banana extraction DNA lab. But again, there can only be so many amazing labs. Are there any last minute questions before we begin our lab? You guys got this. Goggles, gloves, lab aprons, go ahead. I know sometimes they go MIA. Everyone gets so excited to wear these aprons. Good?
I got gloves.
You got the good gloves? Amazing. Here's your lab apron. Ladies, don't forget to tie your hair back. Again, we are working with chemicals. It's important to follow lab safety protocols. Extra apron. I'm sure I can find one around here. You want me to? Yeah. You're welcome. Yeah. I see that it only has one side, but you know, this is what they call using the scientific method. Oh, I was going to bring out how I tie them. Okay. Love this lab. All right, so as soon as you guys have all your lab safety equipment on, you are more than welcome to start following directions. Again, how important it is to follow directions. A procedure is like a recipe.
Remove the masking tape. Or rubber dam.
I got all my safety equipment.
Alright, first up, I don't want to get into the
That's dubious.
Okay, so the cone is going to be on the side like the black electric.
Let's rip tape off the tape first.
Wait, leave it. Black electric.
Yeah, start.
Yeah, we have to make sure that the black stripe is at the end of the
So now we gotta wiggle out the cone.
Your name on it.
make sure.
Oh.
Don't forget lab safety. Teachable moment.
Do not throw
Protect those eyes.
Okay, so now we just put the whole thing in, right?
Yeah. Yeah, so you don't have to take off that.
Okay, just in
Yep,
there.
so you're just going to gently place it between the two plastic divots on the side. Do you see them over there? There you go. And it might be easier to grab it this way.
Okay.
And then gently lower it that way, okay?
Yeah, I think
There you go.
easier.
So much easier, right?
There we go.
There you go.
There we go.
Excellent.
Awesome. Okay, so what's up next? So we position
Position the gel box
on the lab bench so the wires can reach it.
Okay, so yeah,
Yeah, because we have to.
it looks like it already, right?
Yeah. Don't plug it in yet.
Alright, so I think that's positioned well. Not yet. Teacher will come around slowly, add buffer to your.
Gel box, we're pouring buffer into the chambers of
Into the gel if necessary. Okay. We just have to wait for her.
Okay. I think she's getting the buffer right now.
Are we supposed to put this on eventually?
Yeah.
There you go. All right, so we've added the buffer. What are you guys going to be doing next? What's your next step?
Make sure a
We're
pipette
going
tip
to
is firmly attached to the micropiece.
Okay, so we're going to be using our micropipettes.
So we're not plugging this in until after we get everything.
Not yet, because you read the procedure, right? So yes, so you don't want to have any of this set up, right, until you make sure that the DNA is in there. Because if we set it up, it's going to start the electricity.
All right.
Okay,
Right?
so I need to do
So
number
you want to set it up
one.
first.
Okay.
So wait, is it at 10
Colors to the pipette tip.
Yes. Okay.
This is so cool.
Is that just more of like the same liquid?
It's just more of the same liquid.
Okay, now, load the samples to be tested. Make sure a pipette tip is firmly attached to your micropipette. The pipette tip may go on the micropipette all the way. So here, why don't you attach the tip?
Okay, it's on.
Set the micropipette to 10 microliters if necessary.
I believe
It's already
set.
set. There you go. Load the DNA from microtube 1 into the pipette. Only press the pipette button down until you feel the first resistance.
Yeah.
So here.
Just leave it in the.
Wait, so remember, look at the diagram. Here's the.
And then just put it back. Should we open all of them and put them in or no?
So one goes.
So one. So put it in the micropipette in.
and then you can
There you go.
extract it.
So we're gonna put it in the micropipette.
Only press the button down.
Load the DNA from micro in the leftmost well. To load the DNA to the well, make sure you have your elbows on the table for support. Get down close to the chamber and look at the chamber from the top and the side. No, leftmost.
It's firmly attached because remember it's not going to go all the way on like we talked about. Looks firmly attached. All right, so what's your
Okay,
next
so you
plan of
need to
attack?
set it to 10. Is it already on? 10 microliters.
Yeah, it's on 10 microliters.
Beautiful.
So now you have to load the DNA from microtube 1 into the pipette.
Okay, so there you go.
Number 1.
Yeah.
This one's number 1.
Okay.
I mean, open this.
Yeah.
Good teamwork.
Okay.
You could take it out of that plastic if you want. There you go, to make it a little easier. Perfect.
So then,
It looks like actual blood.
first resistance, don't push it all the way down.
Amazing.
Okay, load it in the leftmost well.
Okay, so now remember we talked about this, because you want to have steady hands. So remember you're good. There you go. And I'm going to squeeze in. So feel around for the sides of the well. It's okay that some's coming out. That's perfectly all right.
Yeah, and then just push it through.
And then what's his next plan? Someone help him out.
You need to.
Load the DNA from microtube 1 in the leftmost well. To load the DNA into the well, make sure you have
Is it in?
Yeah, it's in.
Okay, and then what?
And then.
Press the okay.
Carefully stick the tip into the fire as well. Okay. Please stick the tip
And
into
then
the fire.
if you're all done, you can dispose it.
Now, did you push the DNA out? All the way down. There you go. There you go. Okay?
Okay.
dispose of
So,
the tip.
now dispose of the tip into your disposal. Now remember, pop it off like we learned. Perfect.
And then use a fresh compostable
All right,
pipette
next
tip.
person.
So all right.
That is amazing job loading. Amazing job loading.
We have a question.
Yeah.
I'm just curious if it's in the right way.
Yes,
The green
let me
ones
just
look
check.
good.
No, so some of it is going to leak out, and that's perfectly acceptable. There is some in there, yes.
Okay.
Yes,
So
you are
all right,
good.
good. So now we're doing number Jackson.
Now you're disposing of your tips,
Yeah,
right?
just disposed.
Beautiful, excellent. Because again, we don't want to contaminate each suspect's DNA. We want to make sure we know who the culprit was.
That would make
Okay.
sense.
So this is number 3, right? Yeah.
So this is suspect number 3, who is Mr. Farnan.
So
Now, if you want to hold your wrist with the other hand, there you go.
That helps a lot.
Amazing.
So now we just close that in here. All right, whose turn?
So, Sophia.
Sophie.
Alright. And we're doing suspect number 4.
Make sure you're
Now
pushing
number
all
four.
the way down when you're releasing the DNA to make sure that all that micro sample is released.
Okay. Number 4.
So yeah.
Number 4.
Who's that? Nope, that's perfectly fine because you can still see that the DNA sample is still in the well. Okay. So it's still going to travel through the gel.
Okay.
Okay, we were just.
3 your turn. Number 3.
And you're disposing of your pipette. Excellent. Don't want to mix up that DNA.
That one?
Yeah, the one that's open. And then if you, yep. Good control.
Oh yeah.
Yeah.
All right, release it.
This is the crime scene.
Just going to gently push it back together. So just be mindful of that as you move on, not to kind of accidentally pull it against the side of the well. Okay? Excellent. Good job.
Good job.
Okay.
And shaking.
And then you push it down all the way.
Is it okay that our gel is like?
Yeah, because it just then the buffer, so it's
That's okay. That's going to happen. Okay, and that's why we make sure we use enough DNA that it will travel through the gel.
What are we on now? Number 4?
So this is the same like substance as the ag.
Excellent, excellent. And if you want real control with your hand, there you go. And then grab your wrist.
Could you please scoot over?
Very nice.
It says careful.
Then you press down all the way.
Oh!
All right. So that's okay because remember we talked about the fact that this is a very, very soft substance. Okay, so what I want you to do is you have suspect 1, 2, and 3. Okay, so I want you to use the last 2 wells to do 5 and 6. So actually I want you to dispose of that one. That is 5, so dispose of it and get a new sample.
We're just repeating all these steps again.
Ah, dang it, sorry
Stuff like this happens. That's because we're learning how to use these lab techniques, right? So you want to make sure that you have control of your wrist and your arm. So fortunately we have enough wells that we had a few left over that you can use the empty spaces. So I want you to do number 5 and the second to last one. Okay, and straight down. There you go.
Sorry for destroying this.
Okay, and release it. Oh. Now, when you had pushed it all the way down, you were pushing against the bottom. You want to be just a little off the bottom. Now release. Push all the way down. There you go. There you go.
It's working.
See, and that's perfectly fine because again, that's the reason we have extra wells. You guys are great. Keep going.
You ran into an issue.
An issue?
The agar.
Okay, so what I want you guys to do is, again, this is the same thing that happened over there, is that it's a very soft substance. So taking a look, what I want you guys to do is, we have some extra gels that you can use to load. So what I want you guys to do is, I want you to do, let's see, you do have suspect 3, so I want you to do 1, 2, and the crime in the last 3.
Okay.
Okay? It's okay, it happens. This is a learning experience.
Okay.
Right? It is a learning experience. That is
1, 2,
okay.
and
2 in the crime.
Oh, I think some other.
Excellent. Okay. So let's double check everything. We plugged the cord into the back of the power supply. What's my next step?
Then you're going to turn on the power switch in the back of the power supply.
Okay, so again, before we even turn on anything, red to red, black to black. So we have the DNA traveling from the negative to the positive side. Again, why did we have to set it up like that?
Because DNA is
DNA is negatively charged.
Is going to pull the negatively charged DNA.
Excellent, I love it. So then we have to turn the power supply on. We have power. Use the constant button to select V for constant voltage. And we are set for approximately 150 volts. So we are set. Okay, we're gonna press the start button. We are good. And then hopefully, if this is set up correctly, you can come down and see closely. Do you see the bubbles?
Yeah, the bubbles.
Okay, that's because it's in a buffer. And the buffer is charged, allowing us
That the buffer is present.
that proves that the buffer is present, also proves that electricity is traveling through this, because again, we can't see those electrons, but this is evidence that we can see that it's moving. So now you guys can work on your pre-lab questions on the next page while you're waiting. So I would time it for approximately 15 minutes. Okay?
What do we do after 15 minutes?
Read the procedure and you tell me. All right, good job, guys. Should we be able to see this?
Our gel, the very top, when we were putting it in, it sort of split off.
Okay.
We moved it back, but we're just wondering.
That's okay, we'll see how the results come out. Again, this is all a learning experience, and I just wanted to give you guys the opportunity to participate in this gel electrophoresis lab because biotechnology is such a hot topic, and there's so many jobs in biotechnology. So we'll see how it turns out. Okay? So are we ready to plug in and get an energy?
So do we
Okay.
put it in just like on one side, or does it?
Okay, so you want to make the positive goes to the positive, so red, red,
And then that's in
black and black.
right here.
Yup.
Okay.
And then we want to make sure the black side
Plug
goes
this cord
to
in. Nice.
the black side, the red side to the red side. Again, why did we have to set it up this way? If we know that the black side is the negative electrode and the red is the positive, why did we set it up this way?
To create a
Electrical current.
Electrical current, and why? Is DNA...
DNA is negatively charged because of the O negative on the phosphate. So
So of course we want it to be attracted to the positive side.
That's how it'll move.
Excellent. So
in now.
plug it in for me. Thank you, Blake. Okay. And then I'm going to turn the power switch on.
Okay.
We're good to go. I have it set to voltage. I have it set to the max. And we're going to turn it down just a bit. I don't want it up that high. To approximately 150.
All right.
So now evidence, because remember we talked about the fact that you can't really see those electrons in electricity. But evidence that there is electricity flowing through is if you look closely on the sides of the box, you can see the bubbles.
I see the bubbles.
You see the bubbles?
Yeah.
Isn't that amazing? Amazing. All right, so what I want you guys to do is while the gel is running for approximately 15 minutes, keep kind of checking. I want you guys to work on your background information questions.
All right. So at noon we'll check the.
You guys ready? Yes. Awesome. I love this part. This is where the magic happens. This is the magic. All right, so we plugged it in. Before we even turn anything on, we got to make sure that the setup is correct. So we have our gel, black, negative, positive, negative, positive. We're looking good. All right, so here we go. This is where the magic happens. So again, the way we know that this is working and that electricity is actually flowing through, because again, you can't see the electricity, is if you look on the side, do you see some bubbles?
Not yet.
Okay, so let me double check the wires, because that can mean that one of the wires is loose.
Oh, there they are.
There we go. Are we still going through?
No, it happened for like a second when we first touched it.
Okay. All right.
Well, this is from this time.
So I'm going to turn this off for a second. Let's see.
Yeah, it uses the electrical generator force.
These wires are very fragile.
Which forces the Good idea.
Oh wait, don't we have to like? When do we have to like? Oh, because once it like gets to work, wait 15 minutes.
All right. There you go. Let it run. I'll be right back.
Okay.
Okay? Awesome. You're ready? we will make it work. Again, this is all a learning experience, right? I just wanted you guys to participate in this, so don't be hard on yourself. We're learning as we go. And what's nice is we are going to do another lab with regards to this, and now we'll have all the biotechniques under our belts. All right, so here we go. So we're at the next part, so we're ready to set everything up and run.
Yes.
Okay, so we have everything plugged in properly. We're plugged in. Awesome. All right, so let's see. So again, perfect. Can you see the bubbles on the side? Remember we said that was an indication that electricity was indeed passing through.
That's so cool.
Yeah.
So cool.
So this side is the negative current, and this side.
Well, you
no,
tell
this is
me.
the positive.
No, this is
This
the
is the positive
and
side,
that's
so
the
you tell me,
negative side.
because you know this.
Because the DNA is since the DNA is negatively charged,
It's gonna
the
go-
positive side.
You want it to be attracted to the positive side, because that way you'll separate the DNA fragments based on the size, and then we'll get that DNA fingerprint, and then hopefully we'll be able to see who the culprit was. All right, guys, good job.
Thank you. And so, okay.
Oh, go ahead.
We wait a certain amount of, it says in here though, right?
Yep, so you're gonna wait approximately
15
15 minutes, so take a check on the clock, and while you're doing that, you guys can start answering some pre-lab questions. Okay? Good job.
Thank you.
We get
Okay, so guys, at
Let's answer the pre-lab.
11:05. I mean 12:05.
Sound good? So starting Western gel electrophoresis is used to compare everything, maybe compare DNA.
I worked miracles.
What'd you do?
I worked miracles.
She's a miracle.
I'm a miracle worker.
Okay, so now we can start these analysis questions.
Explain the technique of Jello.
Okay.
You guys set up and ready to go? Yeah. All right, so let's double check.
Yes. Gel electrophoresis.
Close this for you. Okay, so again, we have everything good. We're set up. Negative on the negative side. Positive DNA is going to go in which direction?
this way.
What's that way?
That's the positive side.
That's the positive side, because we know that DNA is negatively charged. All right, so we're good to go. And again, we talked about the fact that you can't really see electricity, but evidence is that through the buffer, you have your bubbles.
Bubbles.
All right, so approximately how long are you guys going to let this run for?
15.
15 minutes. So what could you guys be doing while you're waiting? Your questions. You got it. Great job.
They use it when they need to sort DNA strands according to
They don't fit because they're bigger.
What do you think would have happened if you could hit the lousy
Moving. So we have about 10 more minutes left, and then we can.
So you have about
take
10
it
more
off and
minutes
see what
left.
happens. Yeah.
Take it off, and now so you know how to gently slide it off, right? To
Yeah.
take a peek, because you don't want to move this around as much. So you'll definitely slide it off and take a peek and see how it is. You'll see if your bands have clearly separated, and then if so, call me over and I will turn it off.
Okay.
Okay, and so based on the properties, you guys look like you are on track for answering questions. Good job.
Yes. Thank you.
Okay.
So why did you say the longer strands move more slowly?
Because it's remember
Beautiful. That's so amazing. Don't you just love this? Like, this is exactly what they do when you watch your crime scene CSI shows. When they take genetic evidence and the DNA, and that's why I said you guys are going to be DNA technicians today, is that this is what they would do for an actual crime scene, is they would take samples of DNA from different individuals, and because we have these genetic differences, we have different DNA fingerprints. And then that way you guys are actually in the midst of solving a crime. You're trying to see who stole the key to the city. Who stole the key to the city?
But this seems so simple. I feel like in real life they actually have to go through so many steps.
Of course, because
Doesn't it take them a lot longer?
it does. It takes them much longer because, I've given you the samples of DNA.
Oh.
First they have to collect the samples of DNA, and then they have to fragment the DNA with the restriction enzymes. And so, yes, it is a much more complicated process. But if this is stuff that interests you, just like we talked about with the banana lab and the extraction, and this stuff interests you, this is definitely a pathway you guys can go on in the future. Not that I solve crimes one gel at a time.
One piece of Jello at a time.
Yes, it is like Jello. You're right. All right, guys, keep up the good work.
Possible to do this with Jello? To do it with Jello?
All right, how's it coming? Let me take a peek at your gel. So again, we said this is definitely a learning experience. So you can see it is separating. Okay, so it is a little foggy, because this does steam up, but when you get the time and you want to check, you can slide this box off, like this, okay? So you will be able to check it. I don't want to move it around too much because I don't want to disrupt the DNA. So can you remind me again which ones we have in here? So this is 1, 2, 3.
5 and 6. We're only
Actually,
missing
4 is there still too.
Oh, really?
And then 5 and 6. So you guys are saved. You made
No,
it.
I almost completely ruined everything.
So what did we learn from that though? What are we going to do next time we use micropipettes?
Be a lot more careful with gel. That's really easy to break.
It's really easy to break.
Shouldn't they try and make something that's a little bit harder? Because, I mean, wouldn't that help them? Like,
Okay,
I can
so-
just see a crime scene investigation. Okay, here we go. Oh, dear.
And then that's the only sample of DNA that you have? So maybe that's your calling in life.
And then I'm also gonna say DNA.
To not be a DNA technician.
To not be a DNA technician, but maybe to come up with a gel that's more stable.
That side's negative, so.
That's your calling in life, okay?
Alright.
All right, keep up the good work, guys.
And that's negative.
So it's attracted to the positively charged
It's
electric
actually
current.
still working, guys. Yeah. See, don't feel so bad. Just a learning experience. You guys still have your banding pattern and your fingerprints showing up. It may be a little more difficult to decipher, but we'll do that together when you guys get your results.
So to explain how like DNA moves through the gel, could we explain that like DNA is negatively charged, therefore it is attracted to the positively charged electric side, and it's small enough to move through the gel towards the positive side?
Yeah, because remember I said the gel's porous, and those DNA fragments kind of weave their way through those holes. But which fragments are going to move farther?
The shorter ones.
The shorter ones. They're smaller. They have less nucleotide bases to kind of weave their way through.
And is the negatively charged electric current there too? Does it repel the DNA? Is that
Yes,
why?
because like repels like.
Okay. So
Right?
they both like work together to push it.
Excellent. Yes. Yes. Good job, guys. You're on the right track. All right.
That one, since it was like overpowering, it may have broken into it.
Okay, so again, I see this is all learning experience, definitely something we can do. So your DNA fingerprinting, your bands are coming out great still. So again, you monitor it. We want to make sure that when the time comes, you gently slide the top of the box off this way. Don't pull it with the cords. But I mean, you have your DNA fingerprinting coming out.
I think we have about 10 more minutes, right?
All right, 10 more minutes? Beautiful. So when do scientists need, you were talking about this, and what did you guys come up with? When do scientists need to use gel electrophoresis? What are we doing in this lab?
To compare DNA samples to see if they was the right.
So crime scene investigation. Love those CSI shows.
Luke also said that we can use it for like genetic disease.
Genetic disease, that's a great one. I didn't even think of talking about that one yet, because we haven't talked about genetic diseases yet, but that's a great example.
Identifying if
Two different species are related. Really, how closely are we related to that banana that we're actually 50% related to, as we've learned? All right, so again, keep up the good work. And then you guys have a nice drawing and everything, and I could see that, when the DNA goes from the positive to the negative, that it works out. Okay?
Okay.
Okay. Beautiful, beautiful.
Miss Wirkus?
Yes, sir.
So right here, is it possible to see a single strand of DNA? Do you mean like without a microscope or just like?
Without a microscope.
Okay.
When we did our DNA extraction lab.
Yeah.
Could you see that single strand? No.
It has to be clumped together.
And do you remember that fun fact that I gave you about how many strands of DNA you need?
Yeah.
For 1 millimeter, 500,000.
Because a DNA strand is 1 nanometer wide.
2 nanometers. You're
2
close.
nanometers.
So close. You're right. All these fun facts. And this is why DNA is just so amazing. I love DNA. All right, guys, keep up the good work. You guys are doing great.
I think we only have guys, how much time do we have left?
We have 5 minutes.
Beautiful, beautiful.
We were just commenting on how is all of the primary colors that were coming from it.
I love my primary colors.
Is it because are they coming from the different colors that we had put in from the beginning? So it is related.
So again, would I have loved to use real DNA? Of course, because that's, real hands-on experience. But this is actually charged dye.
Oh.
So, but again, if you want to become a DNA technician in the future, you do get to work with real DNA.
That would be fun.
So, or you could be a bio teacher.
We could use the strawberry DNA.
You could use the strawberry DNA, which you'll never look at in the same way again.
Did you keep it?
Yeah, did strawberry take it home?
No, I threw it out. I just squished it around a bit and threw it out.
But you can like identify different types of strawberries to
Well, you could probably identify like strawberries.
What if we did this with
like genetically modified?
All right, guys, how much more time left?
like 2 minutes about it.
All right, the excitement is building.
ready.
Who is the culprit? Who do you guys think the culprit is based on our background?
Based on I think Jody.
You think Jody?
He wasn't in his office.
Yeah, he wasn't in his office.
He wasn't in his office.
Which and they said it was uncharacteristic.
Yeah.
So.
Yeah, that's true because we do always find Mr. Dodig in his office
He's
when
always
we
right.
need him. So that's a good idea. But again, DNA is one of the most profound pieces of evidence, right?
Yeah.
So we're really gonna see what happens. All right, so call me over when you guys are ready.
Okay, we'll do. So, for question 7 on the back, what would happen if you decrease the voltage of the electric current in the gel? What do you guys think?
The excitement is building.
You're talking about where the glass steamed up.
Yeah.
Oh yeah.
It's also
'Cause
used
the
in-
buffer and the electricity going through it. Oh, fascinating. It's like a little waterfall.
DNA is also used in finding the relationship between different species.
Yes, DNA is also used to find the different relationship between species. Like, we're 50% genetically related to a banana. You and I are 99.9% related. So somewhere in our distant past, we were totally related very closely. Isn't that a little scary? Like, we could all be like, we're all related.
Yeah.
Is that scary?
A little bit.
Thanks, guys.
Miss Wirkus, is this like enough differences?
They have more nucleotides. So longer strands have more nucleotides, which makes it harder for them to travel. Yes, it takes longer for them to pass through the pores of the gel.
Okay.
Excellent.
Travel
I love it. How much more time do you guys have on the clock?
We have
5
5
minutes.
minutes left.
Beautiful. All right. You think you're ready? This is the moment of truth. Let's see, who is the culprit? Okay, so you guys took a peek and you feel that the Band-Aid has separated?
Right, Jackie?
Yeah.
I would say so. All right, so again, turn off the power source, number 1, and then we'll gently slide the box off to reveal our gel. And then who's going to tell me what the next directions are?
After approximately 15 minutes, check the status of the band. If the bands have clearly separated, to have verify your teacher ready to move on. The teacher will turn off the current to the power source if you are ready. So check.
Check.
Carefully remove the lid from the gel box. Check. Lift the gel casing tray out of gel box. The gel is slippery, so keep the tray level and gently hold the gel to keep it from sliding out of the gel tray. And place the gel on the light background surface. So here's one piece of paper.
Beautiful.
Provided for you. So we'll document our results after that. So who wants to carefully remove it and put it on the
All right, so let's bring the paper over.
Just put it right here.
Okay,
All
I'll
right.
let you guys work your magic.
Go ahead, Ethan. Be careful. Remember, keep it level because we don't want it to spill.
You got it. Good job.
Go ahead.
Beautiful.
Very nice.
So let's take a look at our DNA fingerprinting. Beautiful. Okay, so here. So maybe the easiest thing is to mark down who is who up here. Right. Right? So you can then determine numbers.
So can someone read to me the order? So it's on the
I'm actually DNA fingerprinting.
So number 1.
I will totally take a picture with my gels and my DNA fingerprinting and how much we match. How much time do we have left?
We're done.
You're done. You're ready? Okay, so you took a peek. We have beautiful gel. Excellent. Love DNA fingerprinting. Okay, so we're gently going to slide the box off. All right, and what's our next plan of attack? Look at, oh, my God. Oh.
Wait, that one on the end with that one and like those two. Wait, a lot
Okay,
of
well, so now before we start jumping to conclusion, what's my next steps? Shades. They're different shades, so we gotta see what's our next plan of attack. I see Sawyer has the paper, so he read the procedure.
Place
the gel tray onto the piece of white paper.
Okay, so are you the man? So you're gently gonna lift. You might want to use 2 hands, one.
Ooh, the water's warm.
electricity.
Electricity.
Oh, and that like the electricity.
If you had your hand in there when
Yes,
when
that
the
kinetic
electricity
theory.
was running, would you be electrocuted?
No,
Only
because
if you directly touch the wires, probably. Beautiful. Okay, so the easiest way to probably decipher it is to relabel the wells at the top, so you could then associate who is with what. Now, Kelsey was saying, yes, they all look the same. They all look like they have the same DNA fingerprints, but really you have to look at the width of the bands, the shade of the bands. Okay, so I'll let you guys kind of analyze and let me know what you think.
So which one's so similar because 99.9%.
Okay, let's
99.9%. Are you guys ready?
It's just a comparison.
It's not Dodig?
It's the second one.
How is it?
All right, are we ready?
Yes.
I think we are. Dun dun dun. The moment of truth. All right, so what's our next plan of attack?
To gently lift the and carefully.
Right, who is going to do that?
Okay, I guess I am.
All right.
Oh, that are
Ooh.
used for each specific thing. So like in eyes.
Peace fell off. That's fine.
It's okay.
We can still sleep.
You got it. Good job. So my best argument for you is, and again, we had our snafu in the beginning, is to label the top what the culprits and the suspects and the crime scenes were. And that'll help you kind of differentiate.
Okay.
Okay, and I can actually, looking at it, I can see. It's still there. It's good. How you guys
Would
doing?
you like to label it?
Pretty good. We only have 30 seconds left.
30 seconds. All right, maybe I'll just hang out unless you guys are almost ready.
We have actually 15 seconds left.
Oh, okay, so we're both ready.
Miss Wirkus, for number one, can we just like explain kind of like the procedure
Yes,
we
that's
use?
all I'm asking. Yep. All right, guys. Moment of truth. Who do you guys think the culprit is? Based on the information.
Suspect 2.
You think it's suspect 2? Who's suspect 2?
Mr. Francis.
You think it's your principal, Mr. Francis? Oh,
I think it's Mr. Dodo.
I think I'm gonna
Yeah.
have
I
to
think
tell
it's
him
Mr.
that.
Dodig.
I think I'm gonna have to tell him
But we
that.
go either way.
He might be a little offended that you think he's the suspect. All right, guys. So now, because we broke off part of the gel accidentally, I want you guys, when you take it out and put it on the paper, to label the top with your suspects.
Okay.
Okay, we'll go from there. Good job, guys.
All right.
All right, here we go. Who do you think the suspect is?
I don't know. Can't
You don't know. Well, what did you think based on the background information that we got that we read?
You said you wanted pretty much all.
You think all of them? You think all of our administrators were in charge?
Just like
Any idea? What do you think?
I don't know.
You don't know?
I would say Mr. Dodig.
You think it's Mr. Dodig? Oh my goodness, you guys. What if I told Mr. Dodig that you automatically thought it was him? You might be
Actually, it's
offended.
not Mr. Dodig. It is.
Well,
Mr. Farnan.
let's take it out first before we start jumping. So I want you guys to follow the next procedure. When you take out the gel and place it on here, I want you to number the top like we did again, so that way it's easier for you to differentiate the different DNA fingerprinting patterns.
Okay, I'm not taking it out of there. Because I
He's
don't
like,
want
I'm
to
not jumping at the chance to take it out. All right, guys.
Do you want us to use colored pencils?
I would when you get there.
We're gonna blame Francis.
Okay, so hold on. You don't want to start shouting it out to other people. All right. Based on the DNA fingerprint patterns, who's the culprit? Great job. You guys did a great job. Who would have thought that Mr. Francis did this? And
I thought it was Dodig.
Yeah, we all
See,
thought
but
it was
now
Dodig.
you could see how DNA evidence can make a situation more accurate, right? Because you have all these scenarios, but like actual DNA evidence is almost undescribable. Like you can't compare it to anything else. Like you could have an alibi. You could say I was here. You could have this, but DNA evidence. This is facts. This is facts. This is facts. I love it. Great job, guys. Great job.
In different pieces.
All right.
So we were wrong, actually.
You were wrong.
know.
Well, who did you blame to begin with?
We thought it was Mr. Dodig. Number one.
So you blame Mr. Dodig.
Yeah.
You blame Mr. Dodig because of the circumstantial evidence we have.
Yeah.
But what does this tell you about DNA?
office. That DNA can go like above and beyond what people see or hear.
Exactly. And then DNA, you can't argue with DNA.
I mean, but it's like, it's almost, they're very close.
Yeah.
So I think maybe if
They're very
we
close, but
further
that
in the
goes
future.
to the fact that maybe if you did have two that were very close, that you could use different restriction enzymes.
Yeah.
And then do it over again. Like
There are
if
multiple
and then you could run the
possible,
gel again. 'Cause there- So, but again, based on this, that DNA fingerprint, which is unique to you, Sorry, Mr. Francis. You know, he selfishly wanted that key to the city.
Yeah.
All right, great job, guys. Okay, continue with your work. Amazing work.
So, number 12, the role of the restriction enemies.
All right. We may have a little difficult deciphering this. Oops, excellent. I love taking that evidence. Okay. Were we able, with what we had, to figure out a fingerprint?
We're thinking number 2.
It's similar fingerprints on here. Suspect 2. So who was suspect 2?
Mr. Francis.
Francis. He must have really wanted the key to the city. Who did you guys think it was in the beginning, out of curiosity? So my whole point is, circumstantially, we said it was Dodig because he wasn't in the area. But then DNA evidence, you can't refute.
Maybe he was working with the parents.
Maybe he was working with a parent. So now we are all going to have to go apologize to Mr. Dodig for blaming him. All right, keep up the good work. Great job. I love it.
Which color has the smallest molecule?
All right.
It's Mr. Francis.
So let's take a look. So what creates these differences, first of all, before we even talk about who committed the crime? The buffer splits the DNA. Now, why does DNA travel differently?
The different lengths of the DNA?
The different lengths of the DNA, and why is it that each person has a different length of DNA fingerprinting? What do we use? Those special what? What's in the buffer? Those enzymes. Those restriction enzymes. Remember, everyone has that 0.1% difference. And it cuts the DNA in different locations, and you all have different DNA fingerprints. So again, when you take a look at it, you can see. So I know, and even though you guys accidentally knocked off the end, it still works. I mean, it was on your side here, ladies and gentlemen, but the evidence shows that DNA fingerprinting says that Mr. Francis. So we're going to have to go have a talk with Mr. Francis and what he's doing with the key to the city. He must have wanted those sapphires. I mean, and every girl likes diamonds, right?
Definitely.
All right. What do we think?
Suspect 2.
Suspect 2, Mr. Francis.
And I would believe that his charge is positive because his went the furthest.
Okay, so his went the furthest, but what does that mean about the size of the DNA fragments?
That they were small and very positive.
They were small and very positive. Well, all DNA has the same charge, but they were small. The restriction enzymes cut those DNA fragments in different ways.
What direction does it migrate
All right.
on the gel?
Ladies and gentlemen, can you all bring it in for just 1 minute? I know you guys are super pumped about answering your lab analysis questions. So the whole point of this lab was to demonstrate, 1, how amazing DNA is. Again, I'm not biased or anything. But 2, I know, but 2, the technique of gel electrophoresis. Okay, I love that you guys have the opportunity to participate in such an activity. And I hope that this incites some passion in a field of biology. Again, I won't be offended if you don't become a biology teacher like me, but there are so many different fields out there that I want to expose you to. So as a result, I heard that many of you actually initially thought it was Dodig, based on the circumstantial evidence, but that DNA evidence, those DNA fingerprints, which are unique to every single individual, proved that who was the suspect? Mr. Francis. Mr. Francis, and I guess we're going to have to bring him up and have a talking to about the key to the city. All right, again, so what I want you guys to do is you're going to finish your analysis questions, and don't forget to follow lab cleanup procedure properly. Okay, so get back to work. Great job. All right. What did you guys think about this experience?
It was interesting.
It was really interesting.
Yeah. I actually have a question about
Yeah.
gel electrophoresis. I remember in the reading we did last night, it said it's relatively new. So, like, how widespread is this technique used? Like, is it used in every crime? Or?
Not every crime, but, crimes where you have to, determine different DNA suspects, because maybe beyond what we have. What is interesting is DNA fingerprinting is new in the sense that it's around 1988, which again to you guys seems new because it's older than you. But yes, it's used for many, many different crimes. So they have them at, you have your forensic station for police work and detective work. And what's interesting is they're opening a lot of cold cases that don't have any, solutions or resolutions to them, and using the DNA that was present and then finding the suspect almost like 20 years later.
Okay.
So that's pretty cool. But again, as we talked about, you're always shedding your skin cells. You're always shedding hair. So keep your DNAs to yourselves, guys. Great job. I love it.
Okay, so what could possibly be the third?
There could be, okay, so we have time stamps and the turn.
It does match. Yours came out amazing, guys. I actually don't want you to dispose of yours. I don't want you to dispose of
Yeah,
yours.
we definitely do.
I am absolutely in love with this Joe electrophoresis.
Yay, ours is we also got the best strawberry DNA.
You did have a passion for that strawberry DNA. So, but again, this is a great gel. You guys really used your lab skills. Excellent, amazing. Following directions. It's like a recipe, following those directions. All right, guys, keep up the good work. So again, don't dispose of that even though it says it in the directions. Great job. Washing off the tray. So again, as you're following cleanup procedure, you may clean up, take off your gloves, goggles, and lab aprons, and then of course you may go back to your desks and use the rest of the lab period to work on your analysis questions. As you know, the lab will be due on Monday. If you have any questions, please don't hesitate to reach out to me via email or Schoology.